And if I go back to the thumbnails view and let's say select some images from, again, this portfolio, just to demonstrate, I can select an image which has, let's say, 2 channels here, and another image which is also 2 channels from 2 different data sets. It's fine. Okay. And now I can go and open with OMERO.figure. So coming back to what I highlighted some 3 minutes ago, OMERO.figure is this great tool for creation of quick publication figures. And if I do so, selecting those 2 images from 2 different data sets, open with OMERO.figure. And then I have a new tab opening in my Firefox, and I have those 2 images in the environment where I can sort them out and create an OMERO figure and a publication figure from them. So what the OMERO figure actually is? Well, it's a plugin for OMERO.web, obviously. And as I'm doing that, you can do that with me. And it fetches the data on the fly from OMERO. So it remembers which images are contained in those 2 panels. This is not somehow disjointed. It's completely connected to OMERO, and in any time it can come back to OMERO and ask for metadata pertaining to those 2 images. So I just made the images smaller, and now I will do copy and paste and paste. This is a usual trick. I showed you a taster of this already in the OMERO iViewer, where we were doing the split view, okay? And now I will make it a little bit bigger. And now I'll do from the first column, I will do a green channel. From the second column, I'll do a red channel. And for me, it's here almost invisible. So I can change the rendering settings even in OMERO figure to adjust them to see much more in those images and in the red channel as well. Okay, very good. And this will be my merge. Excellent. Okay. And now I can see that the images have a very small object inside them. So I can select I can select them all, and I can zoom in. You see the object jumped in. So this is the centriole, obviously, and I need some adjustment. I need a little bit different adjustment from on the top and on the left. So I can just select the bottom and I will adjust it like so. And then I select the top row and I will adjust the top row like this. Very good. I still think I need some adjustment in the green channel here. Okay. And in the red channel. Okay, very good. And then I can go and add some labels on the top of those images to signify where they are coming from and some such. This would be done by selecting the top row and then selecting the channels in the labels tab at the bottom. I will select a bigger font for them. 18, let's say. I don't have to worry about the color. They will pop up on the top here because that's what I want. If I select it like that, here you go. So the channel names are CDK something something and NED1, and this is the merge. Okay, so that's done. I had no idea what the channel names are, but OMERO figure sorted it out for me, and I can now add to the left, I can add the dataset name. So there's some metadata encoded in the dataset name. In this case, these are the names of the proteins which were stained for the images in this particular dataset. Okay, and I will pop it in the left vertical and again 18, and it will appear here. I will say add. Indeed, these are 2 different proteins because they are from 2 different datasets. Great. Having done that, I will nudge with my keys on the keyboard the thing to the left, and I would like to create an insert here. So first of all, it's a good idea always to add some labels. So I know if I go for nanometers, something like 500, and then I say show labels. I wanted to say scale bar, of course, that's what I did here. So the scale bar is added as easy as that. Again, the metadata, the calibration of this image was asked by OMERO.figure from OMERO and was instantly delivered for that image, and OMERO.figure stamped the scale bar on the image. Now I will copy that image and paste it with the intention to do a crop. I can do then in the preview on the copied image, I can simply zoom in. Yeah, and it looks like a crop. Okay. Now I can retrieve regions of interest, let's say on the original image. Now, first of all, how to finish off the crop workflow. If I copy the region so that I have an insert here, of the cropped area in the original image, I can copy it from the crop and then click on the original image, go to labels and say paste. Did you see how I did it? So I have now the insert of the cropped region on the original image, and now I can let's say add some regions of interest. I will do that on this image. I can fetch the regions of interest from OMERO. Ultimately, these regions of interest are coming from Fiji. Okay, so this one is a good one. I want this. Yes, fine. I say okay, and the region of interest is there. It's a little bit too dim, 3pt, and I will make it, yeah, magenta, why not? Okay, so you can see this region of interest has been added here. Magenta is too funky. Maybe let's go for yellow. Oh. White was in the end the best, and I will make it a little bit thicker, 5. Okay, great. So this is a little bit over the top, but it shows the point. So I just added the region of interest, which is coming from the Fiji segmentation. You can see that it's fairly sensible on this one, and I'm kind of happy with this slot. But you know that in the scientific publication, it's very often that one figure contains a mixture of data from very different imaging or even other modalities. So let's just try to add images from some other imaging modalities from a different data set in OMERO into this figure. How do I do this? I go back to OMERO.web client, and I can now find us these conventional microscopy data which we were playing with at the beginning of the workshop, and these are in the siRNAi HeLa dataset. And if I now let's say filter by rating, as I was showing to you 2 hours ago, I can see that some images were filtered out like this, and if I select some of them, then I can say, okay, these are the best ones, and I will maybe go and open them with OMERO.figure, but this would open a new OMERO.figure for me. Instead, I want to add them to the existing OMERO.figure. How do I do that? So I click on the chain icon on the top right, and a link pops up. I will copy that link, and then I go to the tab where my OMERO.figure is living, and I will select the Add Images button. This will pop up a dialog. I paste my link to those additional 3 images from OMERO to OMERO.figure, and I will again sort the pane here. The 3 images are selected now, and I will snap them to grid, make them a little bit smaller, and go through the same routine such as I did with the previous batch. This means I copy and paste them maybe 4 times. Then I select this for this being a blue channel, this being a green channel, this being a first red channel, and this being a merge. That's fine. I will adjust the first red. That's better. And I can now again add some labels. So I'm adding the labels from this time from channels again on the top. First of all, yeah, DAPI, FITSI, GFP, INTSEMP, and the merge. And I can add some on the left vertical. This time I would choose, let's say, the key value pairs. Okay, so if I go for key value pairs on left vertical, I add, and it says animal. That's less interesting. But what about yeah, temperature? Okay, so I need to select it because the label is a little bit. Okay, so you can see temperature is different for all three of them. That's great. And still, I think I'm quite happy with the layout. I can add also another label according to tags. I think if this would be left vertical, I add, and you can see these are the mitotic phases, but the temperature is different for each of them. So anaphase has 37, prometaphase 40, and metaphase also 40. Okay, so you can add really a lot of metadata from OMERO, and OMERO.figure always goes to OMERO, asks what the metadata are, such as tags or key value pairs, and pops them onto your image as labels. Now I can go and adjust the Z section of this image if this is a multi-Z section image, such as this one. And I can go through the Z sections even here, or indeed I can make a projection. So basically, I think that you can start to understand that this is basically solving the horrific workflow which a scientist typically does from a software of interest, such as Zen software, where you have to export the image, let's say as JPEG from the original image. You have to select first the correct plane, then export this plane, put it into Adobe Illustrator. By the way, nothing against those two softwares. Yeah, Adobe Illustrator or the Zen are great softwares, but the problem is once you disjoint the workflow chain between those two, as you do when you export stuff plane by plane and then put them into your figure, and then later find that you can't work with that particular Z plane, you have to go back to the Zen software, export another Z plane, and this will lose you tons of time, and of course a lot of working effort. Whereas here, whilst you are still in a very late stages of the preparation of your figure, you can very easily double back on yourself and correct all your mistakes by simply sliding those sliders. Good. So having done that, I think I will just go back to the labels, and you can see that even post hoc, if I select those three images, I can see that the labels are simply too big. So I can go and make them smaller. Let's say 12. Yeah, and this one is also 12, and yeah. So you can see I could make those labels smaller even much later. I'm happy now. I will add the scale bar as is a good practice. 10 micrometers should be fine. Scale bar is there, excellent. And now I can go and save the figure. I will make a nice name such as I2K-Janelia, and I will see the name of the figure popping up here. Now the URL here in my browser has changed when I did the save action. And this is quite significant because I can grab this URL, copy it, and send an email to my fellow scientist, which would like to work on that figure as well. If they receive that email and they have this link in that email, they can simply click on that link in the email, and this will open a new tab in their browser. And if they are logged into OMERO, then they will be straight seeing this figure of mine. If they are not logged into OMERO, they will see a login screen. After they log in, then they will be immediately re-navigated to this link, which obviously contains this figure of mine, and they will be able to see this figure make comments, make their own copies of that figure, and continue working on that, and some such. Furthermore, I can export the figure to send it to, let's say, scientific journal by clicking the export PDF button. So the figure is being exported right now. And on the server, a PDF is created, and I can now download this PDF. And on my local machine, I can open it with Preview. This is a program for viewing images and PDFs, and it's already happening. So this is already not on the server. It's here on my local Mac in my flat. And I can see that I have obviously the figure with the two panels. And this is not surprising, but why do I have a 2-page PDF from 1-page figure? Well, this is because I have a kind of a metadata page added to this figure page. First of all, the first link is pointing to the OMERO figure itself. So that's the link I was suggesting to send via email to my fellow scientist. So I can click on it in this PDF, and I am jumping directly, because I am logged into OMERO, I'm jumping directly to that figure in my browser. Secondly, typically, when you go and send something into a scientific journal, the process can be quite long, and then they come back to you and say that you need to change panel B on figure 7, and you don't know anymore what are the original raw data underlying the panel B in figure 7. If you go to OMERO figure, you click on this link, you can immediately see exactly what are the images which are used in that figure. So it will bring you and open a new tab in your browser, and the OMERO will find it for you. Very good. Furthermore, if you go to your OMERO figure and see, that's not where I was, but doesn't matter. See the top left, there is a file menu. If you click on that, you can open from that file menu, let's say, and browse the figures of your colleagues. If you have the right to see the figures of your colleagues, I mean, I am Irene Joliot-Curie, obviously, and I have just one figure, that's the one which I just saved. But I can go to the portfolios of my colleagues with their figures and go to trainer 2. I know that my colleague trainer 2 has one figure, but it's really worth it. And I open it. And this more or less showcases what everything can be done with OMERO figure. I can create a multi-page figure. I can do a legend here on the bottom. I can draw new regions of interest, such as these arrows here were drawn post hoc in figure. This would be on top of those regions of interest, which I showed to you how to get from OMERO, which were ultimately coming from the figurre image analysis. So I can draw such arrows. Here is an insert on a very large image. So this is this 100,000 by 50,000 pixel image. So OMERO figure can handle that as well. If I have my Western blots as PNGs and import them to OMERO, nothing stops me to put them into my OMERO figure. As well as, let me highlight the feature of this flexibility of OMERO figure and the changes which can be done on the fly, even post hoc, on things like time lapses here. So you can see that I obviously have 2 time lapses with a sampling, let's say every 40 minutes. And I now realize that this time point, I don't want 42 minutes. I want some later time point. I simply select this, or I can indeed select both of those images. And then I can slide the slider to the right, which will go to the later times. And did you see it changed the labels from 42 to 57 minutes, because now I'm simply highlighting another time point to which I scrolled in the preview panel. And because all the labels are, so to say, live, they are connected via OMERO.figure to OMERO, and OMERO knows about what is the delta t between the time point planes, as well as how many time point planes did I pass through when scrolling, etc. OMERO.figure can ask OMERO on the fly and change the values as appropriate, both the image displayed in the viewports as well as the label value which corresponds. Okay, I hope that you understand now the power of OMERO.figure. Maybe last word on it. If I have the PDF like like the one which I have here locally, say this one, I can still go and open it and post hoc adjust things like, let's say, fonts of labels in software such as Adobe Illustrator. So as I said, nothing against Adobe Illustrator. I can still open the PDF and these things will be vectors. And I can, let's say, choose the font Calibri instead of Times New Roman, which is not something which OMERO.figure will do for me. Nevertheless, of course, once being in Adobe Illustrator, I lose this nice connection between OMERO.figure and OMERO. But nevertheless, this would be a nice follow-up on the workflow I showed you. Are there any questions, ladies and gentlemen?